So bacteria are single-cell organisms. They're prokaryotes. They don't have some of the organelles inside the cells, and they're very simple. But at least to me, it's very interesting because as simple as they are, they can do many, many functions. And not just that, they can also be pathogenic to animals and plants and different organisms. But they can also be beneficial. Like, for example, if we think about our human body, bacteria are the ones that are colonizing the gut system. And because of them, basically, we can absorb all the nutrients and digest all the nutrients or all the food that we're eating to nourish our bodies. Bacteria are very, very important organisms. And they're very small. The size range between 1 to 2 micrometers. They can cause very serious diseases. So in plant pathology, fungal pathogens are the ones that are the most abundant. And let's say in a way more important because they're the ones that really cause most of the damage for crop and in agriculture. However, bacterial diseases can be threatening. And one of the reasons why is because there's not that many mechanisms for controlling them. So if you think about bacteria, you need antibiotics to cure it. But at the same time, humans, we also need antibiotics to cure ourselves from bacterial infections. To use antibiotics in agriculture is a little bit, let's say, challenging to get it approved because you don't want to be spraying antibiotics to the environment and then make bacteria resistant to antibiotics. There are not that many diseases caused by bacteria compared to fungi, but the ones that are out there are very, very damaging. And one of the reasons is because there is little things that you can actually do to control them. So of course, they're microscopic. You cannot actually see it unless they're very, very abundant. And in these pictures that you can see in the top one is just the vascular system of this plant, probably either tomato or potato. It was cut in half, and then you stream it, and then you see the streams of bacteria, kind of the ooze of the bacteria just in the middle. And there's a way you can actually see that's bacteria. And then the one is just the bottom of a plate. Because you have large populations of bacteria, you can actually see it on a plate. In this case, I mean, bacteria can have different colors, and you can actually see it on the plate. So they have different shapes. Bacilliform, which are just basically rods, spherical, some spiral shapes, and some of them have irregular shapes. But the majority of the plant pathogens are going to be rod shapes. So in this case, we have Pseudomonas syringae, that is a rod shape, Xylella fastidiosa, Xanthomonas, Spiroplasmas. These ones are the ones that make these coils. So that's a little bit different, but all of them are bacteria. Bacteria interact with plants in many different ways. Of course, it can be pathogenic, but then it can also be beneficial. So the classic example, of course, is going to be Rhizobium, that is going to colonize the roots of legumes and to fix nitrogen. So in this case, they are going to form nodules in the roots, and they're going to induce the nodule formation. And then the plant is going to give carbohydrates, and then the bacteria is going to start fixing nitrogen and providing some sort of nitrogen to the plant. So this is some of the examples from bacteria that are going to be beneficial. One of the things is that how much bacteria is actually needed to either cause disease or be beneficial. And you really need large populations of bacteria. So maybe just a couple of bacteria that are in the environment are not going to be pathogenic, but then you're going to have like population of 10 to the 6 CFU, which is colony forming units, that are required for bacteria to be either beneficial, like sometimes there's some bacteria that are used as biocontrol agents because they produce antibiotics naturally. And also to cause disease, they're going to need to be highly abundant. Because some of them, or a lot of them, are going to die in trying to reach or colonize the plant. But then, like if they're on top of the leaf and they're trying to get through stomata, with the sun and the dry, like if it's a dry day or something like that, most of them or many of them are going to die. But then if you have very large populations, then there's some of them that are going to actually get in and cause disease. This is just basic characteristics of bacteria. So we have the capsule and they have the cell membrane. But in this case, bacteria do not have organelles and do not have nuclear membrane, which makes it interesting because everything that is happening in the cell is kind of spontaneous. You're going to have RNA production and protein translation happening simultaneously in the cell because there is no nuclear membrane. Or the little things like that in the process for bacteria. All of these processes are just happening like instantly. So let's say if we're talking about plant pathogenic bacteria, so if you have a bacteria that enters the plant and it needs the expression of some genes, of some proteins, then it's just simultaneous. Like the gene expression and the protein expression is going to be happening at the same time. So in many cases, in many studies, when you look at gene expression, you are kind of assuming that protein expression is happening at the same time. I'm going to explain a little bit more later in other slides. So of course, the ribosomes are present because those are the ones that actually produce the proteins. And it has the cell wall, plasma membrane. Some bacteria are going to have flagella. Pili also. Bacteria also can contain plasmids. And these plasmids are just circular pieces of DNA that are independent from the chromosome and contain genes that in many cases encode for proteins that are related to antibiotic resistance or virulence factors to cause disease. Very simple. The genetic material is just the chromosome, which is DNA, and the plasmid, that is also DNA. The cell envelope, it has the cell wall, cell membrane, and capsule. You can have flagella, pili, and EPS, and the inner membrane. The cell envelope has 2 to 3 layers. So you have the cytoplasmic membrane, and then you're going to have the cell wall, and then the capsule. Not all the bacteria can make a capsule. But the capsule is made of polysaccharides, which prevents the bacteria from drying out in the environment, and it can also be a virulence factor. This is the characteristic. The cell walls is the characteristic that actually differentiates bacteria into 2 different groups. So you have the gram-positive. I'm assuming that most of you have heard of the gram staining. So you have gram-positive and gram-negative bacteria. And the difference between one and the other, it's basically made because of the cell wall. So in the case of gram-positive bacteria, they're going to have the peptidoglycan layer, which is very thick and big in gram-positive, where it's very thin in the gram-negative. So the gram-negative has 2 membranes. So you have the inner membrane, which is the one that is going to be surrounding the cytoplasm and actual cell, and then you have the peptidoglycan layer, and it has some periplasmic space, which is just basically a little space in between, and then you have the outer membrane. So it has double membranes in the gram-negative, and very thin layer of peptidoglycan, and in the gram-positive, you only have the membrane, and then directly the very thick layer, peptidoglycan layer, that is going to be surrounding the cell. In gram-negative, it's about 10% of the cell, and in gram-positive, about 90%, not the cell, but the cell wall. Peptidoglycan, why is it so important for bacteria, and why is it so critical for the protection of the bacteria? Because basically this is the one that is going to protect and is going to give some rigidity to the cell and everything. So how is it made of? So it's made of this peptidoglycan monomers. So you have NAM and NAG, which is the acetylmuramic acid, which is the NAM, and then the acetylglucosamine. And then these 2 molecules are going to bound together, and then the muramic acid is going to have the peptides attached to it. So this layer of peptides are the ones who are going to be connected to the other chain. So in this case, if you have the murine, which is just basically these 2 together, and then the peptides here, and then you have another one here with the peptides, and then it's going to have another layer here that it's the one that it's going to be binding the 2 sheets together. So it is the peptide cross-linking that is going to be happening between these 2. And then you're going to end up with this layer that is going to be all the cross-linking, and that's all these layers that are the ones that are going to be making the cell wall of the cell, or the peptidoglycan component of the cell wall in the cells. So in the case of gram-positive, this layer is just going to be very, very thick and big, and it's going to, and of course 3-dimensional. But then in the gram-positive and in the gram-negative, it's going to be very thin layer of that, but it's very robust. And then the gram staining on top of the graph, you're going to have the gram. Let's say you don't know if it's gram-positive or gram-negative. So if it is gram-positive, you're going to have the cells that are basically clear, and you cannot really see anything. So then what you do, if you want to do the gram staining, you're going to put a dye, which is the crystal violet. So this is just a purple dye that is going to enter the cells. Once it's inside the cells, both gram-positive and gram-negative are going to look purple. Now, what they do is that after it's purple, then they put the solution with iodine. The iodine, what it's going to do is going to dry out the cell in a way that in gram-positive, the peptidoglycan layer, it's going to dry out, and they say that it has pores. So the pores are going to dry out and are going to close. So all the crystal violet stain is going to stay inside the bacterial cell, because it's just basically going to block everything that is inside that cell. However, in the gram-negative, the peptidoglycan layer is so thin that once you wash it with the iodine, all the crystal violet is just going to be washed out. So this is the iodine is going to lock the crystal violet inside the cell. Then it goes the next step, which is this staining with acetone, and what acetone or alcohol. So what that's going to do is that's going to wash out the crystal violet from the cells. In gram-negative, then that's at the moment when you wash it out. That's when the cells are going to become clear. However, in gram-positive, because the cells are going to be the crystal violet is going to be locked because of the peptidoglycan layer is dry out and it's going to enclose all the dye. Those ones are going to remain purple. And then you do a counter stain with red, which are going to stain only the gram-negative, and then the gram-positives are going to remain purple. So then what you end up is with bacteria that are going to be, the gram-negative are going to be red or pink, but the gram-positive are going to be purple. And that's one of the most important distinctions in bacteria, gram-positive and gram-negative. Then the next thing is the periplasmic space. This is another difference in gram-positive and gram-negative bacteria. So the gram-positive bacteria basically don't have periplasmic space, while the gram-negative do. So in this case, you have the plasma membrane, and then you have a periplasmic space, then the very thin layer of peptidoglycan, and then the outer membrane. And a lot of processes can happen inside that middle, in the periplasmic space. You have proteins that are binding. A lot of the transport is happening. There's a lot of proteins that are going to be transported from the inside the cell to the periplasmic space. And then some of those proteins may be detoxifying enzymes that can be either shipped to the outside or remain there in case some toxins get in the cell. That periplasmic space is going to contain proteins that are going to protect the cell or the cell membrane, the inside of the cell, against degradation. And the inner membrane is just a double layer of phospholipids and proteins that are going to have pores that can help in the transport of nutrients, and it's stabilized by hydrogen bonds and the hydrophobic interactions. And of course, it's the membrane that separates the periplasmic space to the actual cell. These are some of the activities that are associated with the inner membrane, a transport of metabolites in or out of the cell, phosphorylation, cell wall biosynthesis, of course, happens here, phospholipid biosynthesis, secretion of enzymes and other proteins, and segregation of DNA. So the chromosome of the bacteria is attached to the cell membrane, because it's not just flying around the cell. I mean, it doesn't have a nuclear membrane, but it's not just flying around the cell. So it's usually all tangled up, making a nucleoid, but it's going to be attached to the cell membrane. So what happens is when the cell starts dividing and the chromosomes start replicating, it's going to be attached to the cell membrane. So when everything is doubling and the cell membrane starts to expand, it's also going to pull the DNA, the chromosomes apart. So it doesn't have, I don't know, like, what is it, tubulin, the one in humans, like in eukaryotic cells or in humans that actually pull the chromosomes apart whenever you're doing meiosis or mitosis. Well, in this case, that does not exist, of course, but what you're going to have is that it's attached to the cell membrane, and that's what is going to be pulling the chromosome apart from the cell. So that's why those are some of the functions of the inner membrane. Another difference, like in this case, is in the gram-positives, you're going to have teichoic acids that are not present in gram-negatives. So you're going to have the inner membrane, or the cytoplasmic membrane. You're going to have the periplasmic space, which is just basically this space in the middle. The peptidoglycan layer that is going to have the lipoproteins that are going to be attaching the peptidoglycan layer to the outer membrane. And then the outer membrane is very, very thick, and it's going to have, in the outside of it, is going to have LPS, which is lipopolysaccharides. It may have some proteins that are porins that are going to serve in the transport of nutrients or different things from the environment in or out of the cell. It's going to have the O polysaccharides and the core polysaccharides. And those are important in the cell because, for example, in this case, the lipid A portion is the one that is toxic to animals and is referred as an endotoxin. So this is the one that it's in the inner part. But then you have O-specific polysaccharides and core polysaccharides. And these are the ones that are going to protect against some antibiotics, some poisons and detergents, and some of the host defense mechanisms. But also there are the ones that are receptors for phages and bacteriophages, which are basically viruses that infect bacteria. So how is a virus going to recognize the bacteria? It's because of the O polysaccharide or the LPS layer, basically. Like if you just, in very general, just the LPS layer as the one that is going to be the receptor for phages and bacteriophages. And then I just mentioned porins and lipoproteins, that their function. And this is just a diagram that shows the differences between gram-positive and gram-negative bacteria. And I made this table where it shows each one of the components that are present in the cell membrane, and then if they're present or absent, either if they're gram-positive or gram-negative. It's just basically the differences between them. One of the important things is a different component of bacteria is going to be the flagella. They can have one or they can have multiple inside the bacteria. They're polarly attached most of the time. But one of the things is that flagella is just for swimming. To be swimming towards, in general, is towards a food source or away from a toxin or something like that. It's for swimming. Then we have the pili. I just didn't mention it. The pili, which are short hairs, those ones are for attachment. So that's the main difference between pili and flagella. Pili for attachment, flagella for swimming. But flagella are going to be attached very similarly in gram-positive and gram-negative. And at the base of the membrane, there's going to be a ring. And then you have the rod to go through the periplasmic space, and another ring that is going to be attached in the peptidoglycan layer. Another rod, another ring in the outer membrane, and then the hook, and then the filament. So the filament is made out of small subunits that are called flagellin. So flagellin is these little proteins that when they're attached and get all together, they're going to form the filament of the flagella. But flagellin is very, very important for recognitions from plants to the pathogens. So because flagellin is only present in bacteria, what's going to happen is that in many cases, the plant cells are going to have receptors for flagellin. So they can recognize flagellin in the cell membrane and then trigger a defense response because flagellin was present. And that's what it's called a MAMP. We're going to go over later a little bit more when we study the response of the plant. But I just want you to know that flagellin, which is present in the flagella, it's one of those components that are going to trigger the defense response from the plant, because the plant is going to recognize that it's not plant, that is bacteria, and it has the possibility to be a pathogen. The hook of the bacteria can rotate both ways, and depending on the way that it goes, it's going to tumble, or it's going to have like longer runs or longer swims, and that's important for redirection of the movement of the bacteria. So the way the flagella is assembled is just basically they put the ring first that is going to be hollow in the middle, and then it's like saying, okay, they put it in the membrane, and it's a ring, and it has a hollow, and then it's like, think about each protein that is come in is just put like next to it, and it's just like assembling from the inside out. In the middle is a little empty space, like a little rod, like a little tube, and then it's just assembling from the inside to the outside of the cell until it, like all the components and the flagellin and everything, that's how it's assembled.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rhodobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell's surface, make up the three parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains two mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the mote B proteins. This binding causes a conformational change in mote A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fli-F come together in the plasma membrane to form the MS ring. The Fli-G proteins assemble under the MS ring. Fli-G, along with Fli-M and Fli-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mote A and mote B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but mote B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
In Darwin's Black Box in 1996, Behe spotlighted and made famous a number of really interesting discoveries that had been occurring in biochemistry and cell biology over the last 2 or 3 decades. And what biologists, molecular biologists, cell biologists, microbiologists have been discovering is that at the level of individual cells, there are little tiny examples of nanotechnology, little tiny machines at work. The Flagellar motor is the one that Behe made most famous. It's a rotary engine that powers a whip-like tail, a protein tail that functions like a propeller, and it moves the bacterium through liquid, enabling the bacterium to essentially track down its food supply. And this little machine includes a rotor, a stator, a drive shaft, a U-joint, bushings, bearings, and a whip-like tail that functions like a propeller. And the machine in some bacterial systems turns at 100,000 RPMs in one direction and can reverse direction on a quarter of a turn and turn 100,000 RPM in the other direction.
Bacterial flagellum is a true nanomachine, about 40 nanometers in size. It's amazing. I mean, E. coli, salmonella, which are kind of our model systems for the bacterial flagellum, can propel a cell about 20 lengths per second through a very viscous medium, like water, to these organisms. And you extrapolate that to human scale. 20 body lengths per second, 6-foot person, you know, times 20, 120 feet per second. Mark Spitz or Phelps would be setting records with this type of propulsion.
It's hardwired into a signal transduction circuit that allows the bacterium to sense changes in the sugar gradient in the surrounding liquid.
This signal transduction system is actually a short-term memory system where the cell is, if it's going in the direction of an attractant, a nutrient that it can use to metabolize, it follows that chemical gradient. If it's a repellent, it will sense that and move in the opposite direction. So it's more than just this engine.
It's an extraordinary piece of nanotechnology. It's high-tech in low life. And so just by spotlighting these extraordinary pieces of nanotechnology inside cells, and the flagellar motor wasn't the only one by any means, Behe, in a sense, opened up a window for people. He opened up the black box of the inner workings of the cell and said, look, this is much more complex than anything that the early evolutionary biologists had envisioned. Darwin knew nothing of this type of nanotechnology in cells, and at the very least, we've got to come up with an explanation for this.
And then here's pili. It looks very similar to flagella. The only difference is, of course, these ones are smaller. They're very thin. They look like little hairs all around the cell. Usually are used to attach the bacteria into surfaces. There's a different type of pili, which is the sex pili, that it's going to be found in many gram-negative bacteria. And the idea is that it's used for conjugation, and it's a way of transferring DNA from one cell to another, or plasmid. It could be plasmid or chromosomal DNA that can be transferred from one bacteria to another. And again, this is a way for proliferation of antibiotic resistance genes. And this is just the basic structure of the plasmid. This is the TI plasmid. I will go over it next week whenever we talk about plasmids and agrobacterium. But all of them, they have an origin of replication. That's where the plasmid is going to start replicating inside the cell. And they replicate independently from the chromosome. And some plasmids are going to have some genes that they encode how many of them they can tolerate in that cell. In some cases, there is only a couple of plasmids that are going to be present in each cell. But then in some other cases, and there can be 200 or more plasmids inside one cell. Okay, just as an example, when we were reading that paper that we said, okay, this protein was put into this plasmid, and then it was expressed in E. coli. So that plasmid that is present in E. coli is going to have probably 200 or 300 copies of the same plasmid inside the cell. So whenever you express the gene, you're going to have 300 copies of that gene expressing at the same time in the same cell. And then you have a culture that has millions of cells, and each one expressing so many copies of the same gene. That's why you have so much protein accumulated in that case, and that's why those plasmids are used, or we use it for that purpose. And again, replication is just bacteria reproduce by binary fission, and the only way that they can get genetic exchange and acquire new genes and stuff like that is the conjugation via the sex pili, the transfer or acquisition of plasmids, bacteriophages that can also integrate into the bacterial genome, also mutations and transduction. Transduction, what it is just a cell dies, it bursts, the DNA pieces are flying around, and then a bacteria comes in and it's like, oh, DNA, and then just bring it in and incorporate it into their chromosome, and that's what transduction is, and it's a way of getting new genes into bacteria.